fascinating field. And I'm not going to bother you with a lot of boring words, but we're going to jump straight into the science. And I'm really glad to introduce our first speaker today, who is Professor Thomas BRABLETZ from the University of Nuremberg. And for those of you who are interested in tumor evolution and more specifically in the metastatic field, I'm sure you know his work. He is a pioneer in the different processes involved in this evolution, especially in the identification of and characterization of metastasis initiating cells, as well as, of course, EMT and MET. So for those of you who don't know his work, please go to PubMed. But before, we're going to listen to his talk today. And thank you again, Professor Brablets, for having accepted our invitation. Thank you. The stage is yours.
So, good morning. Can you hear me? Can you hear me?
Yes, yes.
Yes, so much.
Okay. So first, I would like to thank Manuel and Sophie and all the organizers for bringing together this webinar. Of course, Daniel Louvar for inviting me as one of the speakers. So I'm really honored to be one of the speakers in this really exclusive meeting. And although it's a pity that it couldn't be in person in Paris, I'm happy that we can meet here in Zoom, or what is it, Teams? Yeah. I know that I miss, as always, a great dinner, which is a real pity in France. But yes, nevertheless, I'm very happy and to see a lot of friends. Alain Poussieux, hello, and Cedric, and later also Bob Weinberg and Hans Klebers. So it's really nice to gather here. So I will try now to open my presentation, which is a bit tricky because we have a special firewall here. Yusuf, can you help me?
Yeah, open the presentation.
Open.
Yes.
Yes, and now here.
Click on it. Present. Yeah. Yes. Go back to Teams.
Here.
Yeah.
I'm almost done.
Share.
And here again.
Yeah. Ask me to see the cursor.
Okay, can you see the presentation? Okay, as Annick already mentioned, we are interested in cell plasticity and metastasis. And why metastasis? Of course, you know that up to 90% of all cancer-associated deaths still today is due to metastasis. Until now, there is no specific therapy. And of course, to change this, we have to understand the underlying mechanisms and the biology behind metastasis. So, I told you that metastasis is, of course, a major cause of death in cancer. And when we started working on metastasis about 20 years ago, this was the current concept, as you all know, which is due to a stepwise accumulation of mutations in oncogenes and tumor suppressor genes. But already here, from the important step from carcinoma to metastasis, they wrote, the authors at this time, other genetic alterations. And I think till now, till today, there is not a single mutation known that drives carcinoma to metastasis, so the important step. And we thought about 20 years based on very simple observations in differentiated cancers, which are very common, that this might not be necessary, a driving mutation. We realized that here typical well-differentiated cancers, which are known to metastasize often, that they have, of course, are heterogeneous, and they have regions in the primary tumor where this differentiation gets lost towards isolated cells or small clusters. Nevertheless, the metastases are differentiated. And we proposed that this is due to a similar process than the embryonic epithelial mesenchymal transition, EMT, but it can only be transient because it is reversed in the metastasis in the sense of an EMT. And so we proposed that a plasticity of these cells is one of the important features necessary for metastasis. And so cell plasticity and not an additional mutation is likely a major driving force towards metastasis. And of course, you know, EMT is a developmental program, and as it turned out in the last 20 years, it's important for plasticity, but it's only activated in a transient and partial way in most cancers. So in most cancers, cancer cells never reach this final pure mesenchymal state. And you also know that EMT is activated by transcription factors, like twist from the twist family, from the snail family, from the CEP family. And we focused in the last 15 years on factors of the CEP family, particularly on CEP1, which is known or was described as a transcriptional repressor of epithelial genes. And the reason was, or one reason was, at the beginning when we studied these cancers and we did immunohistochemistry, we saw that indeed CEP1, a mesenchymal factor, is activated aberrantly in cancer cells. But if, again, you have a more close look, you see here the colon cancer differentiated in the central regions and the invasive regions. Again, in the central regions of the tumors, in these clusters, CEP1 was not expressed in the nuclei, only in the stroma. But if you look at these invasive cancer cells, you see a clear upregulation of this transcription factor, which should not be expressed in epithelial cells. So over the years, the last 15 years, the work of ours and other groups showed that CEP1 is a crucial factor for all important traits in cancer, which is stemness, plasticity, finally metastasis, but also therapy resistance. And one important finding of us and some other groups was the link to a group of microRNAs, a family of microRNAs, the miR200 family, where CEP1 and miR200 family are strongly reciprocally linked. So CEP1 suppresses miR200 and vice versa. And the one side, the miR200 side, strongly induces differentiation in the same cell, and if CEP1 is dominating, it induces all these important features. So we think that this is a major molecular motor of cell plasticity in these cells. And as you know, in particular work from Bob Weinberg and Alain Poussieux in 2008 showed that EMT in general, but also particular transcription factors like twist, can maintain a stemness phenotype in cancer cells. And the same we could show 1 year later together with work of Michael Clark for CEP1. And for CEP1 we could again show that this link to microRNAs is a crucial mode also to maintain stemness in cancer cells, because these microRNAs not only repress CEP1 but also induce differentiation by suppressing the translation of stem cell factors. So knowing this all, I think I also have to show that it's not only relevant in cell biology and in the lab, but CEP1 and many of the other transcription factors also predict a poor clinical outcome. So it has a clinical relevance, for instance, shown here, CEP1 expression and poor relapse-free survival in triple-negative breast cancer. But nevertheless, these very important tumor-promoting features, these factors, also CEP1, snail and others, are very rarely, if not at all, mutated in cancer. And we think this is also due to the fact that they are important for cell plasticity. So there are stages where these factors are helpful, but there are stages where these factors are not helpful for promoting cancer. And so that's probably the reason why they are never mutated. So showing all this in vitro lab data, the question is, what's the role or what's the function in vivo in a whole organism and in the animal? And you know, the role of CEP1 or the role of EMT and EMT transcription factors in cancer in general, but also in metastasis, was always debated. And in particular, some years ago, 2 papers claiming that EMT is not important for metastasis boosted this debate. But we had additional data at this time that showed that that's not true. For instance, the paper of Rabu, Kaluri, they knocked out snail and twist in a mouse model for pancreatic cancer, which gives rise to highly metastatic pancreatic adenocarcinoma, and showed that knockout of snail and twist do not reduce metastasis. We used the same model, or had the same model in use, and knocked out CEP1 in tumor cells, so only in tumor cells. And at first we were a bit disappointed because we didn't see an effect on the primary tumor. But immediately it became clear, not immediately, it took some time, it became clear that the number of metastases and the size was strongly reduced in the knockout tumors, which are always shown in red here. So we isolated cell lines from various tumors, and as you see here, like the primary tumors, the KPC CEP wild type tumors were always very heterogeneous. So within one animal you find tumors that are well differentiated, even completely undifferentiated sometimes, but the most of them are heterogeneous with mesenchymal and epithelial phenotype. But all the knockout tumors were always differentiated. So in fact, the tumor cells, the cancer cells in the CEP knockout tumors were fixed in an epithelial phenotype. And this also reflects the function. For instance, we see a strongly reduced lung colonization capacity of the knockout tumors compared to the CEP wild type tumors. And what was also striking is that when we compare the tumorigenic capacity of all these different cancer cell lines, we saw that the knockout epithelial, the KPC cells from knockout tumors had by far the lowest tumorigenic capacity. But it was not the mesenchymal phenotype, it was the epithelial phenotype with CEP wild type. So these cells could express CEP1, but didn't do it in cell culture. But they had the strongest tumorigenic capacity. And we again postulated that these are the cells with the highest plasticity, so they can make use of these factors if they need it. And to prove this, or the first hints came when we looked at the tumors more precisely. And you see here the primary tumor of a epithelial KPC carcinoma. And you see CEP1 is not expressed in the tumor cells, but in the stroma. And the tumors make nice epithelial glands. But if you look at the invasive areas, first, if we look at the knockout tumors, they look the same. Of course, CEP1 cannot be expressed in the tumor cells, expressed only in the stroma, and they are differentiated. But now if we look at the invasive areas, we see that these tumors, the epithelial ones, they change their phenotype. They become, like shown at the beginning in colon cancer, they become small clusters or even isolated cells, so kind of EMT. And these cells upregulated then CEP1 in the tumor cells. But this, of course, cannot happen in the knockout cells, so they cannot upregulate CEP1. And this is clearly linked to maintenance of the epithelial phenotype all over the tumor. So we think they lost this plasticity to switch their phenotype. And we also checked this in vitro by, for instance, inducing them with the strongest EMT inducer known by TGF-beta. And as expected, if we start with an epithelial cancer cell, after 3 weeks, we have a full EMT. And if we start with an epithelial cancer cell with knockout of CEP1, this EMT was completely blocked. So they are stuck and maintain the epithelial phenotype. And this is also reflected by the transcriptome. And in fact, in this system, 95 of the TGF-beta response signature was depending on the presence of CEP1, or the genetic presence of CEP1. Also various other types of plasticity, and I want to focus you now on a metabolic plasticity. In this example, on the usage of the two main energy production pathways, OXPHOS and glycolysis. And you see here the OXPHOS rate in a knockout and in a wild type KPC cells, or a mixture of 3 cells and 3 cell lines. At first glance, you see they have a reduced OXPHOS capacity, which is not important here. What I want to show is how they react if we block oxidative phosphorylation, and then look in the same cell what's going on with glycolysis. And you see here the wild type cells, they are able to now compensate this block by upregulation of glycolysis, whereas the knockout cells, they cannot react. They are also stuck in their, more or less stuck in their metabolism. And we are currently investigating other aspects of cell metabolism depending on CEP1 expression. In summary of this part, we have shown that CEP1 depletion does not have an effect on the primary tumor, but we see a strong loss of cell plasticity in these cells with a consequence of a fixed differentiated phenotype, of reduced invasion, stemness, colonization capacity, and in summary, we think, in consequence, reduced metastatic competence. So these data, in addition to, for instance, the data of Raghu Kalluri, show that partial EMT and plasticity and associated EMT transcription factors are in fact key drivers of metastasis. But it's clear that EMT transcription factors have non-redundant subfunctions. So there are functions that are different in snail comparing CEP and twist and so on. And that these factors act tissue-specific. For instance, Raghu has shown that snail has no effect on metastasis in pancreatic cancer, but they are, for instance, Bob Weinberg has shown that snail is important for metastasis in certain breast cancer models. And what I also want to keep in mind, EMT transcription factors are also very active in non-epithelial tumors, where per definition the classical EMT cannot take place. Yeah? And I will show you one side project of our group where we analyzed osteosarcoma now. Osteosarcoma is the most common bone cancer, and it's mainly affecting young adult and children. And it's a real very fatal tumor due to big treatment resistance and high metastatic load. And the cell of origins are mesenchymal stem cells or intermediate lineage progenitors. And why did we analyze osteosarcoma? As I said, it was a side effect. We are not really interested in bone, but when we started or analyzed our knockout allele, and we had a SOX2 allele with very early depletion, we realized an effect in limb length, and we realized that they have shorter bones as you compare here to here. And to make it short, what we found is that the bones are shorter because they run into a premature differentiation. So somehow the stem cell compartment is shutting down, and so they cannot grow anymore. And when we isolated MSCs from mice and then treated these cells with a bone differentiating cocktail, ODM, you see that in the wild type situation, these cells express differentiation markers like ALP or late differentiation marker like alizarin red. Yeah? If we now use knockout cells, MSCs, from knockout mice, you see they show a real spontaneous differentiation, and the final step of differentiation is much stronger than the control. So in summary, CEP1 is important to maintain a stemness phenotype in bone development and blocks osteoblastic differentiation. And that was our start to osteosarcoma. We looked in osteosarcoma cells, for instance, in the cell line horse. Yeah? You see this cocktail cannot differentiate this very aggressive sarcoma cell line. But when we deplete CEP1, they start a spontaneous differentiation, and they can also easily be differentiated by this cocktail. And we think this is due to an upregulation of the genic differentiation factor RUNX2, and in fact we could show that SEP1 regulates RUNX2 expression. And then when we looked at osteosarcoma, we saw that many cancers, many osteosarcomas show a high expression level of SEP1. Some did not express SEP1, and this was highly correlated to the differentiation grade. So the very undifferentiated aggressive expressed high levels of SEP1. So we think it blocks osteoblastic differentiation in osteosarcomas and is associated with poor differentiation. This was also associated, so knockdown of SEP1 with reduced invasive capacity of these aggressive cells, with reduced sphere formation and stemness capacity, and importantly also with a strongly reduced metastatic capacity in mouse models. So the question is, why is SEP1 permanently expressed in many of these osteosarcoma cell lines? This question will come soon. I just want to show you the clinical relevance. When we compared these EMT transcription factors, we found that only SEP1 is more or less always upregulated in osteosarcoma and in osteosarcoma metastasis. And only SEP1 expression was correlated with a poor prognosis. So it's clear a difference to the other EMT transcription factors in this disease. And now the question, why is it expression is maintained in these osteosarcoma cells? You have to know that in most osteosarcomas, there is a defect in the imprinted DIO3 locus. So, and this locus codes for more than 50 microRNAs. And if you look in osteosarcoma compared to normal bone or other sarcomas, these microRNAs, due to this loss of imprinting in this locus, these group of microRNAs are no more expressed in osteosarcoma. And what we found is that 3/4 of these microRNAs were predicted to target SEP1, and in fact we could show for several of these microRNAs that they are strong suppressors of SEP1 expression. So if this group is missing, SEP1 cannot be shut off in osteosarcomas. And we have shown that this is due to hypermethylation of the second allele, and of course this offered the possibility to reactivate this microRNA expression by demethylating agents like diaza. And what we could show that in this aggressive osteosarcoma line, like downregulation or suppression of SEP1 expression, also diaza treatment could induce differentiation in these osteosarcoma cells. And this might have implications also for therapy, because we could show that co-treatment of these cells with diaza and the standard chemotherapeuticum doxorubicin led to a synergy in lethality. So we think that diaza can reactivate silenced microRNAs, induce differentiation, and also sensitize to chemotherapy. So this is the summary of this osteosarcoma part, which I don't show. Finally, I would come back to the major group of human cancers, the carcinomas, epithelial-derived tumors, and ask, do all these findings of us and many other groups have clinical relevance? Maybe the most important, of course, does it lead to novel therapeutic strategies in carcinoma and metastasis treatment? And come back, we and others have shown that we have epithelial primary tumors, epithelial metastasis very often, and in between this partial and transient EMT phenotype. And the current treatment today mainly targets the differentiated tumor mass in the metastasis or the primary tumor. But what is missing, or what is persisting, are the green cells shown here, the cells that have often undergone a partial and transient EMT. And this is due that the fact that it's now known that EMT in general and EMT transcription factors in particular can confer also therapy resistance. And I'm sure you will hear much more about this by Bob Weinberg's talk at the end. So the question is, how can we target this resistant state? We could either inhibit the initial EMT, or we can try to shift back the mesenchymal state into an epithelial state, or we could also block the retransition to an epithelial state, because likely or often this partial EMT state is coupled to a dormancy, so a non-growing state. Or we could directly target this highly resistant, till now really difficult to target state. Yeah? And I can say in all three options, or for all three options, a lot of groups are working. I would like to show you one brief example that we have done for blocking this state. And we asked a simple question. We screened a panel of epigenetic drugs with the question, which of these drugs can upregulate these highly relevant microRNAs. And we came up with one HDAC inhibitor called mozetinostat, which was able, compared to other HDAC inhibitors like SAHA, to strongly upregulate these microRNA families. And you see the mozetinostat treatment also strongly reduced expression of SEP1. And you see it also in vivo. If we injected a highly aggressive pancreatic cancer cell line, it's expressing SEP1, is undifferentiated, no ekaterine expression. If we treat the tumors and the mice with mozetinostat, SEP1 is downregulated and ekaterine is upregulated. And then if we treat the mice with gemcitabine, the chemotherapy drug in the clinic, you see in black, the tumors grow. Gemcitabine has no effect because it's a highly resistant cell line. Mozetinostat alone has a slight effect, but the combination strongly suppressed the tumor growth. So we are currently working on other combination schemes. So how much time do I still have? Yeah, a few minutes. So in the last part, I would like to at least discuss a bit how we could target this directly, this highly aggressive EMT transcription factor expressing intermediate state. And the problem is that EMT transcription factors, or transcription factors in general, are difficult to target directly. And all these factors have physiological roles. They regulate basic functions in the whole organism. So are there maybe specific functions of these transcription factors, in this case SEP1, that can be selectively targeted, for instance, in cancer cells? And we were addressing this question. And as I've shown you before, SEP1 is known as or first described as an transcriptional repressors by binding to co-repressors, to e-boxes, and suppress differentiating genes like ekaterine miR200. But we and others have shown that it can switch to an activator in the same cell under different genetic conditions. And we have shown some years ago that it can bind to the hippo pathway effector YAP and then co-activate strongly hippo and TEAD target genes like CDGF, AXL, and so on. And then we went on with this study, and using ChIP-seq data sets that we generated, we looked for additional motives. So, of course, the best motive was the SEP1 binding site. We already knew that it also a motive is the YAP-TEAD binding site, but the second best was AP1. So it was very often correlated with SEP binding. And we could prove that SEP1 also directly binds AP1 factors like JUN and FOS. And then when we overlap the transcriptome or the ChIP-seq peak set for SEP-YAP and SEP-JUN peaks, we saw that a strong overlap. And in fact, we could figure out that all work together to activate genes. And we then analyzed the whole genome and found that there are many genes that only have one peak, we call them SEP1 only peaks. And there are many areas where we see all three peaks nicely overlapping. Yeah? And when we better analyze the underlying or the genes, we found that the SEP-only peaks mainly were localized in the promoter region, and the SEP-June peaks, the triple peaks, mainly were localized in enhancer regions. And what we also found is that most of the single peaks were regulating genes that are repressed by SEP1, and the triple peaks were regulating genes that are activated by SEP1. So in summary, without going into more detail, we have shown that in the same cell, in parallel, SEP1 can act as a repressor, in this case of tumor suppressing genes and differentiation genes, by directly binding to EPOX, or it can act as an activator by building or by being integrated in a huge activator complex, and then activating tumor promoting genes. And in summary, it leads to all the known consequences of these EMT transcription factors. And this we think has clinical relevance. For instance, we have shown that if we see all four factors expressed in cell lines, these are only the Claudin-Low subtype, the very aggressive subtype in breast cancer. And as you know, expression of these factors indicates a real poor clinical outcome, poor relapse-free survival, poor reaction to chemotherapy, and highly metastatic risk. And if we integrate also the major target genes into this calculation, you see again they mainly expressed in the aggressive Claudin-Low subtype, and also the target gene set, the top 10 target genes, strongly predict poor outcome in breast cancer. So that's the situation in carcinomas. But it is interesting that, that's the final slides, it's interesting that it's different in other types of cancer. This group has shown that SEP1 can interact with LEF in glioblastoma. We did not see an interaction with LEF in breast cancer and other carcinomas, and they did not see an interaction with YAP and AP1 in glioblastoma. And it's unclear what's the activating complex in other tissues. So we think this might be a chance for specific intervention. And what we are currently doing is to map the specific interaction of SEP1 in this complex with the idea to then selectively block this function of SEP1 in carcinomas. And with this, I would like to summarize. Many cancers, most important human cancers, are often differentiated, but they can make differentiated metastasis, and we think that in between there is this partial and transient EMT state, which in summary, of course, shows that plasticity of cells is important for tumor progression. And we think that this gives the chance for a therapeutic intervention in the future. And with this, I would like to stop and thank you all for listening, and in particular thank members of my group, which are just in Erlangen or were former members. And I want to thank in particular Nora Feldker. She was involved in the last part with the interaction with AP1, which was based on the work of Waltraud Lehmann, who found the interaction with the Hippo pathway activators. Angela Krebs did most of the KPC knockout work in the mouse models. Simone Maithoff found mozetinostat, and Manuel Roux almost alone worked on our separate project on osteosarcoma. So thank you again for listening, and I'm open for questions.
Thank you very much for this great talk. Maybe I'm going to start with a general global question. Why do you think that so far, despite all the research, stemness, but more globally, plasticity has never been successfully targeted? I mean, at least to go further to clinical trials and so far.
Yeah, of course, that's the most important question, and also I think the direction where the whole field tries to go. And I think there are, as I mentioned, there are many groups and maybe also companies now going into this field and really trying to make use of it in the sense of novel therapeutic strategies. In fact, yeah, as you will see also from Cedric's work, it's a very complicated situation with many subgroups and subtypes and many factors from the environment that are influencing the whole process. And that's maybe the reason why we are not yet in the clinic, but I'm really optimistic and convinced that maybe within the next 5-10 years, we see great progress.
Okay, I hope you are right. Now I'm going to ask you the question on the chat. The first one is from Sergio Roman from Institut Curie. He asked, is ZEB1 overexpressed also in the invasive front of the metastatic setting in secondary organs?
Yes. In principle, at least in many cancers, the metastasis builds up a similar structure as in the primary tumor. And you also in the metastasis, that's what we already published 20 years ago when we only looked at the morphology of these tumors. Also in the metastasis in the rim, for instance, of colon cancer metastasis to the liver, you see again cells that lose differentiation, for instance, upregulate nuclear beta-catenin, and also these cells then often, not always, but often show expression of EMT transcription factors in the metastasis.
Okay, the next question is from Laurent Lecam in IRCM in Montpellier. He said, thanks for the talk. And have you identified ZEB1 target genes which could explain the metabolic phenotype you identified in ZEB1 knockout cells?
Yeah, that's also, of course, a very obvious question. We are currently working on it, and we have some candidates which we have to confirm in further experiments. But they are of course candidates, and they are also known for other EMT transcription factors. So there are many metabolic factors as a direct transcriptional targets of these factors. Yeah.
You have any example or?
Maybe, for instance, hexokinase could be a direct target, and the others we are currently working on. I don't want to tell you now because we are in the middle of the work here.
Okay, thank you. The next question is from Fatima Meshta Gregorio from Institut Curie also. She said, very nice talk. And she said, did you compare glucose, glutamine, and lipid consumption in ZEB1 knockout versus wild type cells?
So what, can you repeat it? Glucose and lipid, what?
Consumption.
Consumption, yeah. Yes, that's also an important question. In the mesenchymal phenotype, or let's say in general, and this also refers to the first or the question before, ZEB1 is known to regulate the lipid metabolism and to regulate lipid consumption in a cell. So in ZEB1 expressing cells, you have an upregulation of certain phospholipids and certain phospholipid producing enzymes. Yeah? So it favors the phospholipid and lipid pathways, yeah, in summary.
Then a question from Alain Puisieux. He said, thanks again. ZEB1 has no effect on the development of primary tumor in the KPC mutant RAS and P53 model, but you also have shown that it does have a significant effect in the presence of wild type P53. Could you comment on that?
What we see in this KPC model or in the KC model with wild type P53, that CEP1, and we don't understand it, affects the very, very early stages, the ADM, and even before. So we have less ADM when we knock out CEP1. But first, we do not know what's the reason. Mark Stemmler in our group, he worked 2 years on it, and we couldn't find a clear reason for this. And the second is, if the cancer grows, this early difference was somehow overrun. Yeah. So when the tumor is grown, you don't see a difference. But in very early steps, we see a difference. We don't know the reason. We would like to know, but maybe if somebody has an idea, he can send me an email. What could be a reason? But yeah. I cannot hear you.
The next question is from Suzanne Sebens, and she asked, what are the conditions defining whether a cell is fixed in an epithelial or mesenchymal phenotype, or whether it exhibits a plastic phenotype? Does it require a certain set of mutation or environmental conditions?
Yeah, that's also, I think, a very important question. Why are some tumors plastic? Why are some fixed in a more epithelial phenotype, so less aggressive? And why are some tumors fixed in a highly aggressive, or not necessarily aggressive, but in a mesenchymal phenotype? Of course, I think the major reasons are additional underlying mutations. Of course, there can be mutations that abolish plasticity, but because they fix the EMT state. Yeah? And it was shown, for instance, in work by Roland Rath, that if you have a high level of mutant RAS, yeah, the cells are likely fixed in a more mesenchymal phenotype. And it also may depend on the signals from outside. If you have a lot of infiltration cytokines that can likely affect the phenotype of the cells, you see a high plasticity. If you don't have these infiltrations, it could be a reason for low plasticity also. Yeah.
Okay, thank you. There's a question of Fanny Jolin from Gustave Roussy. She asked a question about PDAC and ZEB1. If metastases form but are smaller in, I guess, in knockout conditions, does that suggest that invasion is not inhibited, but growth at secondary site is possibly related to a decrease of number cancer stem cells or metastasis initiating cells, or because of the microenvironment? Can you comment on that?
Yeah, again, this is an important question because, you know, the metastatic process is very complex, and it can maybe stop at very many different, or be affected at many different points. What we think is, if we inject the same number of cells in the tail vein, and then we look how many cells go to the lung, we see no difference. Also in the bloodstream, we don't see a difference. But the cells that reach the lung tissue, then they make a different number of metastases. So we think that a major function that is blocked or depending on CEP1 is the colonization process, which itself is a very complicated process, as you know, and includes stemness maintenance, which includes relief from dormancy and all these things. So the precise molecular mechanisms we don't know yet. Maybe there are various mechanisms that are affected by CEP1 loss, but we think it's something around the colonization process, which we did not yet define more precisely.
Okay, another question from Fanny is that when you correlate in the clinical data, when you correlate ZEB1 with the poor prognostic and so on, do you think that it's the involvement of ZEB1 in the stroma or in the tumor part?
Again, pointing out to let's say a big problem when you analyze clinical samples of for AMT transcription factors. Of course, the most if you simply analyze RNA or transcriptome data sets, you must be aware that the most of the expression comes from the stroma in many cases. Yeah? And so in some of the data that I have shown, to be honest, this was not separated. But we also have data derived from immunohistochemistry where we can determine the SEB1 in tumor cells, and also here there are data sets where we and others have shown that the expression in the tumor cell is a predictor of poor outcome. But it's difficult, yes, with most published data. Yeah.
Maybe single cell will help to answer this question.
Absolutely.
I think we've got still 3 minutes, so I'm going to ask another question from Gaël Fortin. Do ZEB1 knockout mice show defects in hematopoiesis and or hematopoietic stem cell exhaustion?
Yes. That was shown by different groups. Also, they also used our SEB1 floxed allele, and they have clear defects in differentiation into different hematopoietic lineages, which I don't know all by heart, but they were all published recently. I think 2 papers in the last year. SEB1 is also important for the correct differentiation into hematopoietic lineages.
Okay, we're going to finish with a question of Pierre Savanier. Hello, Thomas. Nice to see you. Great talk.
Hello, Pierre.
Any evidence e-boxes could be involved in stabilizing transcriptional complex when there is a transcriptional activation?
Also, again, a lot of really good questions, I have to say. We also addressed this, and it looks like, I don't want to say it for all loci that we analyzed or that we see for instance, in the ChIP-seq for SEB1, it looks like that for the activating function, the e-box is not necessary, at least in the context of the complex that we described with TYAP AP1. If we mutate the e-boxes in constructs, it has no effects, whereas in the repressive function, it's absolutely crucial. So this is the tendency or the hypothesis that we have in the moment. So likely it could be that SEB1 does not need to bind directly in the activating condition, but also this is not yet fully proved.
Okay, thank you very much, Professor Brablets. And I think it's time to shift to the talk of Dr. Cédric Blancpain. So again, I think there's no point of introducing Cédric Blancpain from the University of Brussels. He has done a lot this last 10 or 15 years on stemness in normal but also in cancer field, mainly in the skin and the breast. If you don't know, again, his work, please read them, and you're going to appreciate especially all the lineage tracing models that he has set up. And I'm glad then now to let you talk, Dr. Blancpain, and this is your turn. Thank you very much.
Thank you very much for the nice introduction. How